If you start using Cradle, you can kick off a project. You define which protein you're interested in, and you define the objectives that you're trying to meet for that project. Then you can run base models on Cradle. And what these base models will do is provide sequences that they think are plausible, given what it understands about evolution or about previous data sets that you've uploaded. And by testing these in the wet lab, the model now starts to understand really the relationship between specific mutations and the real world outcome. And this enables you to move further towards the desirable outcomes, and also for the model to make trade-offs and find kind of the sweet spots where you are meeting all these different objectives.
Welcome back to The Smart Biotech Scientist. I'm your host, David Brühlmann. While our focus is typically on bioprocessing, today we're looking beyond our usual scope to explore what's happening upstream. We're going to talk about protein design. I'm excited to have Elise de Reus, who is a co-founder of Cradle, the generative AI platform revolutionizing protein engineering. With a PhD in fungal synthetic biology and experience engineering microbes at high throughput, Elise bridges computational and experimental worlds, making biology programmable and doubling protein engineering speeds. Are you ready to discover AI's protein revolution? Well, let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Elise, welcome to the Smart Biotech Scientist. It's good to have you on today.
Thanks, David. I'm excited for our conversation.
Yeah, likewise. I'm very excited. Elise, share something that you believe about biotech development that most people disagree with.
I think biotech can learn from the field of software engineering in their willingness to more openly share what's worked and what hasn't worked. In software engineering, there's a strong culture of open source, building on each other's ideas, whereas in biotech, we are very prone to wanting to reinvent wheels and keeping our best ideas to ourselves, which on the whole slows the industry down.
Yeah, we would love to hear from you, biotech scientists, what you think. How can biotech use and leverage best practices from other industries? I agree. Yes, we can definitely learn from the software industry, Elisa. But before we talk about the exciting work you're doing at Cradle, let's talk about yourself, your story, because you have a fascinating journey from early interest in life sciences now to co-founding Cradle. How did you get interested in biotech, and what were some interesting pit stops along the way that led you now to co-founding a company?
Well, DNA was my early love. I thought it was so fascinating how this molecule was the blueprint for life. And especially when I realized early in university that there are so many applications of biology in the world around us. So taking these tiny cells and molecules into the largest factories producing the chemicals around us or the medicines that we are treated with in hospitals. And a pivotal moment for me after working in labs and studying life sciences in university was actually in working in a dairy industry. So a large dairy company where the challenge that I realized this industry had was balancing all these different components of milk across the whole world, across all the different requirements of the markets, across the seasons where cows produce maybe more fat in winter and more lean milk in summer. And the reality that you can't turn off a cow when you don't need it to produce milk, and you can't get a cow to specifically produce more protein or less fat if you need it to. And also, cows are living beings, which brings a whole range of other ethical and practical considerations. And while I was working in the dairy industry, I heard of a startup that was producing dairy protein from microbial fermentation. And the advantage that has is that if you brew dairy protein without the cow, like brewing beer, but not producing alcohol, but producing, for example, whey protein, you can turn off a fermenter and you can decide where it sits, and it consumes much less land, water, and energy compared to the cow. So rethinking how we produce very important products in the world around us from biology was what really convinced me to deepen my expertise in the field and commit my career to the field.
And what was the pivotal moment that led you to co-founding Cradle?
So my journey took me from doing a PhD on fungi, studying all the molecules and proteins and enzymes that they can produce, to realizing that research and development timelines are long and wet lab work can be slow and it can be tedious, and wondering where in the world that was happening at a faster pace and with more success. And that led me to a company called Zymergen in California, so West Coast, where there were a lot of different types of robotics and automation used in the process of producing small molecules or proteins from biological systems. And when I got there, I was so excited to have access to all these new technologies that enabled me as a scientist to do 100-fold or 1,000-fold more experiments compared to when I was doing my graduate work. But I realized that even then, the success rates of these programs were often still low and the programs were still expensive. And so it was a thought of recognizing, if this is what we can do with automation and this is still the limitation, we need as an industry a next step change to make sure that biotech has the impact in the world that we hope and think it can have. And then I was lucky to meet the co-founders at Cradle and have conversations that ultimately led to starting this software company.
And so now you are working in protein design. Can you tell us a picture of what the current state of art is before we dive further into how you're doing that as leveraging technology? But how do people typically design proteins?
Yeah, so the traditional approach to protein engineering is using often directed evolution, so introducing mutations in a less targeted way, first through scanning where in the protein you could maybe try different mutations, selecting different starting points in nature, and then observing which changes are beneficial and combining those together to ultimately lead to a protein that meets all these different criteria. With tools like AlphaFold, so predictive AI tools, scientists have more understanding of how a specific sequence leads to, for example, specific structure. The next step in this protein engineering workflow with AI is using generative methods, and generating new protein sequences that are better aligned with the final application in the real world.
So what are the current limitations when it comes to protein design?
So proteins are complex molecules, and we as humans have very specific requirements for a protein to work in a real setting and real application. So if you think about an antibody in a cancer medicine, you want that antibody to very specifically bind to the cancer cells and not your healthy tissues. You also want that antibody to be manufacturable, which means it has to maybe be soluble, not aggregate, survive the process from the factory into the patient. Similarly, for enzymes, they may need to be highly active, not produce any of the wrong products. And again, you have to be able to manufacture them and express enough of them. So all of these approaches or all of these considerations, constraints, means that it's very hard for a scientist to rationally engineer a protein so that it meets all these different requirements. Maybe you can optimize the stability of a protein, but you don't want the changes that lead to improved stability to simultaneously degrade its activity. These trade-offs are very hard for humans to make rationally, even with some of the predictive tools that are out there today. And that's where generative AI approaches can really accelerate timelines, which ultimately means that drugs get to market faster or processes can be substituted by like greener biotech-based alternatives.
As we dive now into the generative AI, can you tell us how you develop your algorithm, how you develop, or like what kind of technology are you using for generative AI, and how do you train the whole AI so you can, as you say, reprogram or make biology programmable? Because as I understood, there's a lot of moving parts you have to consider, but that's actually where AI is pretty good at, right?
Yeah, absolutely. Yeah. Science seeks to understand the natural world, right, through observation and experimentation. And in engineering, you apply these scientific principles to design and build and optimize. And for this to work, you need to be able to predict experimental outcomes. And in the case of protein engineering, be able to generate proteins that align and lead to these better outcomes. And so Cradle ultimately makes it easier for biologists who are designing and engineering proteins to do this in less time and with more success using generative AI. Now, what do we mean by that? Generative AI, you can think of it as ChatGPT for proteins, where these protein models have an understanding of the protein language. So ChatGPT understands the protein language. And then you can make these models more specific to the problem that a scientist is trying to solve by helping them to speak the specific language and then the dialect of this project. So you can think of it as adding specific data for the project to help the model become an expert at that challenge, and allowing the scientist to use this expert model to generate proteins that have a higher probability of binding to that cancer cell while still being stable.
Now, traditional approaches leverage computational methods. How do you do that with your generative AI? Is that a hybrid approach, or is that completely detached from traditional computational methods?
So the new workflow for protein engineering using generative AI still involves analyzing the proteins. So, for example, their evolutionary trajectory is very important. It also still involves wet lab validation. So using a generative model does not mean that you get the perfect protein in one go with 100% success rate. You do need to go through iterative cycles where you test the output of this generative model in the wet lab. And you can imagine that with ChatGPT, if it provides you an answer, so you prompt it with a question, it provides you an answer, and you can fact check that answer very easily. If you prompt a generative protein model, that would look like, this is my sequence of interest, my protein of interest, and this is the objective that I'm trying to work towards. So please improve the stability or make it stable at a higher temperature. This model would then provide a set of protein sequences that have mutations. And as a scientist, you then validate these in the wet lab to produce the data, which can feed back into the model to help it better understand the relationship between the protein sequence and how it behaves in the real world.
To what extent is your model capable to understand mechanistic insights? I know AI is very good at coming up with a good answer, but can it also help you as a scientist then to understand, okay, if we add more of this amino acid, I just say something random, I make it up, then we would get a better binding, for instance, affinity with our receptor? Or where are we there now with the generative AI?
So Cradle, if you start using Cradle, you can kick off a project. You define which protein you're interested in, and you define the objectives that you're trying to meet for that project. Then you can run base models on Cradle. And what these base models will do is provide sequences that they think are plausible, given what it understands about evolution or about previous data sets that you've uploaded. And by testing these in the wet lab, the model now starts to understand really the relationship between specific mutations and the real world outcome. And this enables you to move further towards the desirable outcomes, and also for the model to make trade-offs and find kind of the sweet spots where you are meeting all these different objectives.
I'm curious because on the podcast, we've talked multiple times about AI in various ways. And something that comes up very often in bioprocessing is that we don't have enough data actually to fully leverage generative AI or AI in general. How is that in the protein design? Do you have enough data to fully leverage the technology?
Yeah, that's a great question. And in biology, you often see that data sets are short and fat, which is a way of saying you have few different variants and you have a lot of different information about each variant. Now, the cool thing in this era of generative AI for protein engineering is that we're seeing that you can meaningfully update the understanding of a protein model about how this protein works in relatively few data points. So very concretely, that means that if you, as a scientist, want to use Cradle to improve your proteins, you can test just a 96-well plate of designs in every iteration. And a 96-well plate of designs, it's not nothing, but it is within bounds for many different labs in both academia, startups, as well as industry. This is something that is so meaningfully different from the state of protein research 5-10 years ago, where you had to start training models from scratch, and that took tens of thousands of data points. Now, in the new state, these large protein models have such a good understanding of how proteins look in the natural world that they can, seeing very few data points specific to the project, very quickly grasp how to introduce changes to drive better outcomes in your application.
And this is absolutely key now in biologics development, speed to market, because a lot of people are going after the same targets. So it's very important to be as fast as possible. Can you tell us how you are specifically accelerating the protein design steps and how you're going to do that? For instance, let's say I have this new disease target. How would you now proceed and help us as a company get there much faster than other people?
Yeah, so it's being faster to market. So first in class, it's also best in class. So having the best solution, which involves not just cutting out experimental rounds by making bigger improvements round over round or optimizing for multiple properties at the same time, rather than saying, first, we're going to make sure that our antibody binds, and then we're going to worry about whether it is developable and whether it's going to cause unwanted effects once it goes into clinic. So it's reducing times as well as getting to better designs. So finding non-intuitive solutions. And how we see this in projects that we do with our customers, not all of which I can talk about for obvious reasons, is, for example, last year at a conference, we presented together with scientists at J&J a project on developing a vaccine against Staphylococcus aureus, which is a pathogenic bacterium that is associated with 1 million deaths annually, or in 2019. There, the challenge was to stabilize the vaccine while keeping its efficacy. And using Cradle, scientists at J&J stabilized the protein in just one round that took 2 months, whereas a similar project had taken over 2 years and 7 rounds of experimental engineering.
And how do you differentiate yourself? Because a lot of startup companies now doing AI, and probably a lot more are coming into the market every single day or week. So I imagine it's quite a competitive space. So how are you different, and how are you positioning yourself as a company compared to the others?
So we started Cradle because we believe that generative AI is now at a point where it can meaningfully accelerate research across protein engineering, across different modalities and different domains and different industries, and that we need this impact to work towards planetary and human health. We decided to help maximize the impact of this new wave of technologies by making a software platform and operating as a software business, so as a SaaS model, which means that teams that use Cradle pay for using the Cradle software without sacrificing royalties downstream or having to pay milestone payments. It also means that they don't sacrifice IP, so any sequences that are generated on the Cradle platform belong to the users of Cradle. And importantly, data stays private, as do models that interact with customer data. So these pillars of the business model allow Cradle to be a very useful tool to accelerate protein research across different industries and across different customers without having to sacrifice royalties or IP. And then, of course, you can talk about the capabilities. So when our customers evaluate which strategy to use to leverage AI for protein design, in-house, it's very important to ask the questions. Is this alternative that we're considering able to predict experimental outcomes? Is it able to generate improved proteins? Is it able to make trade-offs, so to optimize across multiple properties at the same time? And how does it do this? And also importantly, how are these approaches benchmarked? So at Cradle, we have a wet lab in-house. And the sole reason for having this wet lab is to be able to test the behavior of our models in the real world before any new iteration is released to all of the customers who use Cradle. And I can't stress enough how important it is to be able to observe these real world outcomes of the models in a wet lab setting, because you can't engineer these systems to work reliably and robustly without taking them into the lab. So experimental validation is so important at every step along the way, both in the development of the models and the algorithms, as well as in the final protein engineering projects themselves.
And how will that play out in the future? Do you think at one point we'll be able to skip the wet lab experiments?
I mean, the dream is to get to an ML solution that in a single protein design solves for all the different parameters that a scientist specifies. We are far away from that. But having these new generative technologies, we're definitely closer than we were 5 or 10 years ago. And the cool thing is that having a solution like Cradle means that you can also scale across all these different programs that are running within an organization. And it also means that different types of scientists and computationalists can work together to leverage these machine learning technologies and apply them in the lab and drive these programs towards the ultimate goal.
That's part 1 with Elise de Reus, from traditional protein engineering challenges to Cradle's AI-powered breakthroughs. In part 2, we'll explore real-world applications, impressive partnerships with industry leaders, and practical advice for incorporating AI into your protein work. Do you have biologic process and technology development challenges? Well, book a free consultation at brühlmannconsulting.com and we'll help you accelerate your development timelines and reduce costly iterations through strategic CMC planning. Thank you so much for tuning in. Don't forget to leave a review on the platform you found us on, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.